Hi, I'm Laura Guillardin. I am a PhD student at the University of Oxford, and I'm going to talk about genotyping strategies for large genome conifers and how genetically diverse are UK evergreen woodlands. So this is the outline of the talk, starting with the background. As we all know, the main challenge that we are facing at the moment is climate change, and it's affecting every ecosystem. Talking more specifically about forests, how climate change stressors like extreme temperatures, pest and disease, drought and fires affect forests is incredible. Like it's a huge thing. And if those forests are not adapted or they not have the tools to adapt to these new circumstances, that's a big problem that we are seeing already. On the other hand, there is a willing to change to these multi-purpose forests from even age plantations. So we want to keep timber production, but we will also enhance the landscape, increase the biodiversity. We will transform these forests into more sustainable forests. And how to do this, there is a way to do that. There are many, but one way would be to use a continuous cover forestry approach, which is basically to transform these plantations by making thinnings, clearing, in the end, opening space to let the natural regeneration to appear, and also to let some new species to appear. This could be also enhanced by planting or doing selection thinnings. So we know the story in terms of silvicultural tools. So we know how to get there, but the problem, or what I'm looking at in my project, is if those primary trees that we see at the top, they don't if they don't hold enough genetic diversity, but this approach rely on natural regeneration, how is going to be the genetic diversity of these new offspring? It won't be enough to tackle those climate change problems that we are seeing. Well, basically that's it. So that's very briefly what my project is about. So the populations that I'm looking at are in the UK, and the species are Pseudotsuga menziesi or Douglas fir, Tuya plicata or Western Vetsida, and both of them are from Northwestern America. So here you can see in this map that this is mainly the region where the seed source come to then plant those seeds or plants in Europe for timber production, which is the two of them are very widely used. I would say that it's more used Douglas fir at the moment, although, well, yeah, it's more used. Now I want to show this graph where you can see the different genome size for different organisms. So there are here the conifers, here's the size of the genome. So you can see the big difference between conifers and other organisms. It doesn't matter crop species, model species like Arabidopsis, even human genome are around this size. And our species, Pseudotsuga menziesi or Douglas fir is here, like I think it's 18 gigabytes. And we have here, it's not shown, but here will be the Western Vetsida, around 9.1. So looking at this graph, what you can see is the huge genomes that we have to work with when we analyze the sequence that we get, for example. So this can be a challenge and can be, I won't say difficult, everything is difficult, but I will say that we need to find different strategies apart from other model organisms to get some results and conclusions. So the hypothesis, as I said, is that the planted trees that compose these UK forests may not hold enough genetic diversity, So how the gene pool is being transmitted to the offspring. To answer that, the main objectives at the moment are that, first of all, we want to assess the genetic diversity in the canopy trees and compare it with the natural regeneration so we can see the differences. Then we also want to assess the level of genetic variability across the study species. And then we will compare that with some data that we have from provenance trials that I showed before. And the last objective will be to evaluate these differences in the genetic diversity at different stages of continuous cover forestry. So we will see that. Coming now to the experimental approach and results, I will just focus on the genomic strategies to genotype my samples. So I am starting with Pseudosugamen CSE or Douglas fir, and we chose Fluidium technology to genotype those. This technology is based in this chemistry that I'm going to explain very briefly. One of the advantages that it has is it doesn't need a whole lot of DNA because you can perform this pre-amplification step. So that's very useful. Then it's basically based in 3 primers, one with the reference allele, another one with the alternative, which will, well, that will be in the next slide, sorry. So basically these 2 primers and then the locus specific primer, which is specific for each locus. And then you add this reagent with the fluoroforms. What happens is these 2 primers will get attached and will get incorporated to the DNA fragments. It depends obviously in the allele. And then that will also will call, let's say, the fluoroforms, and that will all get attached. And that will be how the different devices that we need to use will, how you say it will see the sign, the signal, sorry, and then it is when you get the genotyping done. It sounds a bit confusing, but it's not. It's easy, and it's also very fast to do, and you can perform lots of PCRs at the same time. So it's a great approach. We will, yeah, it's a great approach. After, well, so now the question is, how can we get there? How can we develop those primers, right? For our species, Pseudosugamen CSE, we are happy because there is a huge database with more than 28,000 SNPs that Howe et al. Put together from 2 other databases. Sorry. How these SNPs looks like, there is 35 base pairs in each of the sites of the SNP. That's a problem because for this technology, you need a longer sequence in both sides of the SNP, but I'm not going to go into that. What I'm going to talk about is the number of SNPs. So this technology that we are trying to use and the aims of my project, I don't need that much SNPs, right? I need, or I aiming to have around 100 SNPs to then measure the genetic diversity, because I have loads of samples. And the problem now is how to choose from this huge database to that small amount of SNPs. So we select those SNPs by performing different filterings. We first of all, and I think the most important one, we aim to have a high mineral frequency. So just making this filtering, we go to 800 from the 28,000. Then to be sure, I blasted them, and then I got just hits for 322 SNPs. So I keep those. And then I want them to be totally accurate, right? No mismatches, no gaps, well, just one mismatch, which is a SNP, and we get 191. And then we want one SNP per region and one region per SNP. And doing that, we get 170 SNPs, which is more or less around the number of SNPs that we want to try in a sample of SNPs, sorry, in a sample of samples, in some of them to just be sure that they work properly. So that's what I'm doing at the moment. This is just an example of 12 of them with the call rate is not great, but we're getting there. And from those 170, we're aiming to get to work perfectly around 100, and then we will genotype all them. We will genotype all my samples with them, like creating a sort of chip. Now, for the other species, which is Tuya plicata, we are not that lucky. There is genome assembled, there's a reference genome available, but there are no SNPs. There are no molecular markers described yet, or they are not available at least. So what we performed is this pre-step of SNP discovery, and we decided to use genotyping by sequencing, which is a technique that is very commonly used in non-model organisms. I'm not going to explain it a lot. It's just you choose restriction enzymes, and these restriction enzymes will digest the DNA and will create fragments. So then you get the libraries, and those libraries with those fragments is what you will sequence in short reads. So after you sequence those, you will get the raw reads, and you will need to perform different bioinformatic filterings again, a bit more not difficult, but a bit more, well, different than the ones that I just talked about, like the filtering. It's more straightforward, right? And then you will analyze your data. How to choose restriction enzymes? In my case, what I did, because there is a reference genome available, there is a tool which is called DD-RAD-seq tools that allows you to perform a simulation using whatever enzymes you want. So performing all these simulations, we decided to use a PST1 and SP1 pair of enzymes because of the number of fragments that we were looking for appear to be around these numbers. After deciding the enzymes, we sent our DNA to be to prepare the library, and then to be sequenced. And then the next step is basically to analyze the sequence reads, which is what I'm doing at the moment. And I don't have any more results. I could have some more, but because of delays in the reagents deliveries, I'm a bit stuck at the moment. But thank you so much for attending my talk. And I will just have to thank all my group mates, and Ella, which was my field assistant, and yeah, my supervisor, John Mackay, and my second supervisor, Gary Kerr. Thanks a lot to all of them. And yeah, if you have any questions, please feel free to make a question now, or just send me a message, an email. I'm also on Twitter. So yeah, thanks a lot.